Assalamualaikum and hi everyone. This is a video lesson. It is a continue of chapter 8.0 Recombinant DNA Technology. Overview of this chapter. First, 8.1 Recombinant DNA Technology. Second, 8.2 Methods in Gene Cloning. And I believe you already learned about these two subtopics in the previous video lesson. This video will be more focused on 8.3, which is application of recombinant DNA technology. At the end of this lesson, students should be able to briefly explain applications of recombinant DNA technology in mass production of insulin using cDNA. The second, describe the steps in production of insulin using cDNA. Application of recombinant DNA technology. With the advancement of genetic engineering, it is now possible to transfer genes between distantly related species. For example, you can transfer genes from human to bacteria. The desirable genes can be transferred from lower organism to higher organism or vice versa through recombinant DNA technology. The practical application of DNA technology of course affecting our life. There are many fields benefit from DNA technology and genetic engineering. For example, agriculture, forensic, environment in order to stop pollution. And not to mention, medicine. In this video, we only focus on the application of recombinant DNA technology in the field of medicine, which is the production of hormone insulin for diabetes. Additional knowledge for you: the history of production of insulin. This is a short video from my YouTube that will show you how the previous scientists developed the production of insulin using recombinant DNA technology to help the diabetics to live their life.
Diabetes is a disease caused by excessive amounts of blood glucose. This condition, known as hyperglycemia, is fatal if untreated and leads to various symptoms such as excessive urination, fatigue, weight loss, and blurred vision. In order to control blood glucose levels, a hormone known as insulin is made by the pancreas to signal cells for the uptake of blood glucose. Type 1 diabetes is caused by insufficient blood insulin and is believed to be caused by a variety of factors, such as viral infection from the rotavirus and measles and genetics. In 1921, Dr. Frederick Banting researched a successful way to isolate insulin from pancreatic cells in dogs. They later began mass-producing insulin for diabetic patients from pigs, saving many. Porcine insulin and human insulin only differ by one amino acid residue at the C-terminus, in which the alanine in pig insulin is replaced with a threonine in human insulin. Because of this meant that pig insulin was compatible with human insulin receptors. While animal insulins are still sometimes used today, they must be very pure, as there are impurities that have been shown to cause allergic reactions. In 1926, scientists first crystallized insulin, and then in 1955, a breakthrough by Frederick Sanger allowed scientists to sequence the DNA coding for insulin. The 3D structure was solved by Dorothy Hodgkin in 1969 through the use of X-ray crystallography. After Sanger's work, scientists could use this genetic code to synthesize their own insulin. Efficient mass production of insulin began in 1978 through recombinant bacteria, in which insulin genes were inserted for bacteria to transcribe and translate into insulin. Thus, the insulin that you use is mass-produced by lab bacteria. For more information on type 1 diabetes and diabetes treatment and management, please visit the Protein Data Bank website. Thanks for watching.
Alright guys, so this diagram is showing you a rough idea on how we produce human insulin. But before it, let's get the laser pointer first. So we have a human cell here, and we also have a bacteria here. What are we gonna do? we will extract out and cut the insulin gene using a restriction enzyme. And then we also will extract out the plasmid from a bacteria, and we will cut across both strands of the plasmid using the same restriction enzyme that we used to cut the insulin gene. That means we are using the same restriction enzyme to cut the insulin gene and also to cut the plasmid. After that, the insulin gene will be combined together with the plasmid using DNA ligase. Therefore, the plasmid is now called as a recombinant plasmid and is ready to be inserted into a cell or a bacteria cell to be specific. So this bacteria cell will read the gene as if it's one of its own, and it will produce protein insulin. This bacteria reproduce asexually and so very quickly, and you can get a population of billions of bacteria that containing human insulin. So basically, this bacteria will act as a manufacturer of human insulin. Lastly, we will extract the human insulin from the bacteria, and it is ready for use for diabetics. The synthesis of DNA from RNA template via reverse transcription produces complementary DNA, or in short, cDNA. So how it is formed? The diagram on your right shows you a sequence on how to produce a cDNA. But before it, let me have a laser pointer first. In this diagram, a purple zone indicates environment in a cell nucleus, while a blue zone indicates environment in a test tube. In the cell nucleus, we have DNA of eukaryotic gene. We will transcribe our desirable genes, for example, human insulin gene. We will transcribe it into RNA primary transcript. RNA splicing occurs, which means we remove introns and forming mRNA. So this pink strand, we call it as the mRNA. It will be exported out from the cell nucleus and placed in a test tube. In the test tube, reverse transcriptase will use the mRNA as a template to synthesize the first strand of cDNA. After completion of the first strand of cDNA, we will degrade the mRNA using the degradation enzyme and continue with the synthesis of second DNA strand using DNA polymerase enzyme. At the end of this sequence, we will have two blue strands, which means we are forming cDNA without introns. Therefore, it is safe to say cDNA is DNA molecule made in vitro using mRNA as a template and the enzyme reverse transcriptase. A cDNA molecule lacks introns compared to DNA of the genome. Therefore, it can be used directly to express the proteins. cDNA is often used to clone eukaryotic genes in prokaryote because cDNA represents the expression of the genes without the introns. Thus, no process for RNA splicing in bacteria cell.
A messenger RNA molecule has a 3' poly A tail. So, using an oligo-DT primer and reverse transcriptase, a complementary strand of DNA, or cDNA, is synthesized, forming an mRNA-DNA hybrid. The mRNA strand is degraded with alkali, and the free 3' end of the DNA strand forms a hairpin loop that primes synthesis of a second DNA strand using DNA polymerase III. S1 nuclease cleaves the hairpin loop, leaving a double-stranded cDNA copy of the original mRNA.
Production of insulin using cDNA. What is insulin? Insulin is a hormone that regulates the amount of glucose, or we call it as a sugar, in the blood and is required for the body to function normally. So insulin is produced by cells in the pancreas. We call it as islets of Langerhans. These cells continuously release a small amount of insulin into the body, but they release surges of the hormone in response to a rise in the blood glucose level. However, if you are type 1 diabetic, unfortunately you cannot produce the human insulin anymore. You have to inject it for the rest of your life. One of the best examples of the application of DNA recombinant is how bacteria have been engineered genetically to produce human insulin. Therefore, it helps the diabetics to have a continuous supply of insulin and will help them to live their life as normal as possible. The first step is the isolation of our desired gene. mRNA for insulin is isolated from the beta cells of islets of Langerhans in the pancreas. So have a look on the diagram on your right. We have mRNA extracted from pancreas. Now we will locate and remove our desired gene, which is human insulin gene, using a restriction enzyme. The restriction enzyme will cut at a certain end of the mRNA in order to get our desirable genes. After that, we will produce a pro-insulin mRNA. The second step is the formation of cDNA. Reverse transcriptase enzyme is aided to synthesis a first strand of cDNA by using mRNA as a template. If you have a look at the diagram, the green one is the mRNA that acts as a template, while the blue one is the first strand of a cDNA. After that, mRNA strand then discarded by using mRNA degrading enzyme. So we will remove the green strand and then we continue to the next step, which is adding the DNA polymerase enzyme to synthesize a second DNA strand complementary to cDNA in vitro. So here we will have two double blue strands that twist together to form a cDNA, or we call it a double helix cDNA. We continue the step by cutting the cDNA and isolate just the sequence that encodes for the insulin protein using a restriction enzyme BAM-H1. Then, plasmid removed from a bacterial cell is cut at a specific site using the same restriction enzyme BAM-H1. As I mentioned earlier, that the enzyme that used to cut the insulin gene, the desired gene, is also the same restriction enzyme that will cut across the plasmid. We cannot have two different restriction enzymes in this process. We only allowed to have one restriction enzyme. The cDNA is inserted to the plasmid using DNA ligase. The recombinant plasmid now is carrying the human DNA for insulin. Another sequence diagram that shows you the procedure on how to produce human insulin. It is quite similar with the previous diagram on the previous slide. I would like to remind you the three major steps involved in the production of insulin. The first one is the isolation of desired gene. The second one is the formation of recombinant DNA. And the third one is the gene transfer to host. Before we end our video lesson, you are required to answer questions from a quick quiz. This quick quiz won't take up much of your time, I promise. What you have to do is scan the QR code below and answer the questions. Thank you and best wishes. Miss L.